So, hi, I'm Kelly Shanahan, Board of Directors of Mediviver, here again on our virtual C2C because thank you, COVID, we still can't take the RV on the road. And I'm here today with Dr. Heidi Ford. She is from the University of Colorado, and she is a professor in the Department of Pharmacology and the Associate Director of the Basic Research Program. Now, Dr. Ford got a grant from Mediviver way back in 2012, and it was when we were still doing pretty small grants, so really just seed money. But I think that's something that is so important early in an investigator's career to get a little bit of money to get their ideas off the ground so that then they can move on to the big NIH and all these other kind of grants. So thank you so much for agreeing to join us today from your office with the beautiful artwork there in Colorado. And what I'd love to do is for you just to, to tell us, our audience, a little bit about what you did with that research money that we gave you all those years ago.
Great. Thank you, Kelly. And, thank you to Mediviver for funding us with that seed money years ago. It was quite a while ago. I think at the time I was probably an associate professor, but just barely. And we were very interested in understanding, how metastasis happens and what types of molecular pathways contribute to that, with the hopes that we could find targetable pathways. And so the emphasis of my lab is really trying to understand how developmental regulators, so genes that are critical in normal embryogenesis or normal embryonic development, that tend to get silenced when after development is complete and turn back on in cancers. So often cancers don't sort of reinvent the wheel. They hijack mechanisms that are present in cells at different stages when cells are doing things like migrating a lot and invading a lot and proliferating and growing a lot, because that has to happen when embryos are formed, right? Normally those type of processes are silenced, but then in cancers, those things get turned back on. And so what we were trying to do was identify pathways that were highly expressed in embryogenesis, but downregulated in most adult tissues, with the idea that if they were then hijacked by the cancer cell to promote metastasis, that if we targeted them, the side effects would be pretty limited because you wouldn't have expression of these or function of these in the adult, right? And so back when Mediviver funded us, we were working with transcription factors. So transcription factors are proteins that bind to DNA and then actually cause gene expression programs to turn on. So they turn on many other genes, right? And we were working with transcription factors that are really important in normal development. So they're considered master regulators of development, and they're key for doing things like causing cells to massively grow in order to make up enough cell mass for a certain tissue, for example, right, during development. But then that needs to get silenced when development is complete, because once your organs are formed and the cells are all grown, you don't want more. You don't want them to keep proliferating, right, and growing.
Yeah, we don't want to all be giants or with massive livers or things like that.
And so those are the kinds of master regulators that often get turned back on by metastatic cells, because now they utilize. So again, these particular genes were important in things like turning on gene programs that affect migration and invasion. And now we know evasion of the immune system. So things that you might need early that again, sort of get re-expressed in the cancers. The problem was that transcription factors are not very druggable. So we are working on drugging them now, and I can talk about that a little at the end. But at the time, especially, nobody thought you could drug a transcription factor. So while everybody agreed they're absolutely critical for mediating metastasis, nobody knew how to stop them from doing what they do. And so I had a really talented student at the time who thought she would go after something called microRNAs, which are small RNAs that are known to control gene expression, that at the time held a lot of promise for being more druggable. And still, actually, lots of people are working on that. It hasn't been as easy as we had hoped. But in any case, so what she did was she identified a set of microRNAs that were regulated by these transcription factors that we were interested in, and found that they were promoting metastasis. And so what Mediviver funded us to do was to figure out how they were promoting metastasis and then to see if we could identify ways to inhibit their ability to do that.
Right.
And so,
Yeah.
so anyway, so it was a seed grant or a pilot grant that was really, really helpful because it was hard to get a large grant on this at the time because it was very new and we needed to generate more preliminary data. We did end up getting a very large grant on the topic. Again, we were trying to do things like therapeutically, try to inhibit this. And that's a little high risk. So there are a lot of these larger grants that you get from the National Institutes of Health. You really need a lot of preliminary data to take the risk out of it to some degree to get them to fund you. And so this was very helpful to be able to do that. And so we actually did make real great progress on understanding how the microRNAs contributed to metastasis. And what we showed is that, I'm sorry, my phone keeps beeping. I should have put that away. What we showed was that it was important for causing cells to be more stem-like. And that stem-like characteristic, we think, can allow cells to toggle between different states pretty easily. And that enables them to be able to handle all the stresses of the different metastatic environments they'll find themselves in, right?
So for our audience, I just want to, if we can explain a little bit about what stem cells are. So basically stem cells are cells that kind of can do anything, sort of like when we're embryos, because we start with an egg and a sperm that then fuse, and that becomes everything from your hair to your toenails, to your spinal cord, to your heart. So stemminess means cells that kind of get that ability back to kind of grow and do things. And stemminess in cancer is not, it's not good.
Right, exactly. And that your comparison with development is great again there too, because that's exactly what we were looking at, are things that could be important developmentally and normal, but now are being used out of context, wrong place, wrong time, right? So we identified that it affected what we call the tumor initiating capacity, but it's the same kind of thing as stemness. And we actually worked out the molecular pathways that it was influencing to do that. We focused on a couple of key signaling pathways like TGF-beta and Notch signaling, which are known to affect stemness and more aggressive properties in tumors. And then we did spend some time trying to use various means to inhibit the microRNAs in animals, but it turned out that had a lot of side effects. And so it didn't turn out to be, that part of it didn't turn out to be as efficacious as we had hoped, which was disappointing, but actually got us back to, and so now this is years after the Metaviver grants, right? Deciding we really need to go and hit the transcription factor itself. So instead of trying to get the microRNAs, we want to get the thing that controls it. And that is the thing that's very specific to embryogenesis. And in fact, we've most recently, we started a couple projects where we did high throughput screens and identified small molecules that could target these pathways. And we now have 2 different small molecules that we think can target the transcriptional complex that results in activating these microRNAs and actually results in activating a lot of other things that mediate metastasis. And in fact, we just published our first paper in 2020 on one of those small molecules and showed that it could inhibit metastasis. So
yeah, and I think one of the things that I think a lot of people that aren't involved in science or research, you always think that if you don't prove your hypothesis, if what you're setting out to do doesn't work, then it's an abject failure. But just like in life, I think sometimes we learn more from the things that don't work than sometimes we do from the things that do work. And reading your final summary, there were some of your hypotheses, like you thought that TGF-beta was going to be super important, and then you found out, well, maybe like it's not. It seems like this other pathway seems to have more promise. And then, as you just mentioned, you kind of went back to the basics, like, okay, we were looking at this microRNA, but maybe we need to look at these transcription factors, these other things that potentially have more, might have more potential for translational work, for something. Because I see this all the time among patients and among groups. You'll see a headline about something like, new technique, cures cancer in mice.
Yes. Yes.
And I'm like, if I had a nickel for everything that cured cancer in mice, I wouldn't be sitting here. I'd be like on a beach somewhere, and I'm like all you researchers, would as well. I wish things worked as well in humans as they do in mice. So I'd love to hear about, where, that little fork in the road led you down a different way, and what work you're doing now in the realm of metastasis.
Yeah, so we're doing quite a few different things. So the fork in the road, again, that led us more to going back to it maybe convinced us not to give up on what we were trying to do in the first place, which was go back and try to inhibit the transcription factors. It's kind of the holy grail. I don't know how to turn off this phone from ringing. It's kind of the holy grail in cancer therapies, right? So you've probably heard of MYC. If you could figure out a way to inhibit that, that would be great. And so we decided to just do it. Let's just do the really high risk things we were trying to avoid. It also made us realize that while that microRNA, it was a cluster of microRNAs, is really important, it was only one of many things that were being transcriptionally regulated by the transcription factors themselves. And one of them is called 6 and another one is called IA, I's absent. And they're both named for what they do developmentally, actually. But anyway, so we have a whole aspect of the lab now that's working very actively around sort of drug development. Can we take these small molecule hits that we have and make them into something that we can, yes, we'll first test in mice, which we've done to some degree, right? But then move even further. So we've actually even looked at applying for these small business types of grants. So we can actually move these more towards the, from the preclinical to the clinical trials, et cetera. But we're still, I would say, a long ways off from that. But it also made us think a lot about when we go back to the stem cell aspects of this. So the fact that we saw that this was affecting stemness, and then we started to appreciate that it's not so simple that every single cell in a tumor has that phenotype and looks like a stem cell. It's a proportion of the cells, and the tumors are very heterogeneous with respect to who looks more stemmy, who looks more. So in the case of breast cancers, they're very epithelial tumors, right? But we study quite a bit something called the epithelial to mesenchymal transition. And I'd rather refer to it actually as epithelial mesenchymal plasticity. And that is where the tumor cells can move between different states.
Sometimes they go the other direction.
Exactly.
Right.
So they're plastic. It's not just I'm going in this direction to become more mesenchymal, let's say. And that's going to make me more metastatic. It's the ability to move back and forth that's important, right? So we've been studying a lot that plasticity and also the crosstalk between cells within the tumors, some which have those characteristics and are more stem cell-like, let's say. A stem cell is the most plastic cell there is. It can become anything, right? Given the right factors. And the cells that are more sort of stuck in their ways, I would say, more differentiated, right? And how that crosstalk actually promotes metastasis. So we've been really interested in how the cells that are more plastic can talk to those that are less plastic and actually secrete factors that make those less plastic ones migrate more too and move more too and metastasize more also, right?
Kind of like pheromones, to bring it into something that might be a little bit more understandable. Some plants and animals secrete pheromones to attract a mate. And so you have cells that are secreting these factors that attract these cancer cells and encourage them to metastasize, because everybody wants new neighbors. Rather than not be in our bones, liver, and lungs, but
that's absolutely right. So that's been another big focus. And then the last sort of thing we're focusing on quite a bit around metastasis is the immune microenvironment. And there's been a lot. We've also done some work on adaptive immunity, so T cell responses to tumors, but we've now gotten very interested in the responses of other cell types to tumors, macrophages, natural killer cells, more innate, as they call it, innate immune cells. But in all cases, we're trying to tackle different aspects that can affect metastasis in the more complex tumor microenvironment, right? So instead of focusing only on that one cell that has those characteristics, we're focusing now more on the crosstalk with those cells, with different cell types within the tumor, with the tumor microenvironment, et cetera, because it's really a much more complex problem I mean, you have to sometimes be reductionist in order to understand the pathways, but you then have to put it back into the context of everything else that's going on, right?
Right. And all of us would like to see, research that, like, we're doing research that's gonna lead to a drug that's gonna help us. Kind of the way I feel about my car. I don't care how the engine works. It just works. And all I need to do is press the ignition and it goes. And I think that's the patient perspective. But on the other hand, if somebody didn't understand how an engine worked and how all those moving parts fit together, then our cars wouldn't go anywhere. And it's the same thing with research. We have to understand the basics. We have to understand the whys and hows of, why does the cell become cancerous? Why does it decide to leave, the breast and go find another home? Before we can develop the treatments that are going to keep people with metastatic breast cancer and other advanced and metastatic cancers alive longer, we have to understand those basics. And I think that can be really hard for us as patients to accept that. But babies don't come out of the womb running marathons.
Yeah.
you gotta roll over and then crawl and then walk before you can run.
Yeah, I will say another aspect that I've gotten very interested in, and I want to compare some of the inhibitors that we've developed. We know they can inhibit the process of metastasis, but we often get a lot of criticism, even from the scientific community, around that, because there's the argument that, once you have metastasis, inhibiting the process is not going to help that much. So I would make
that's not true.
Exactly, right? I was going to say,
yeah,
metastases concede other metastases, right? It's absolutely not true. But you still get that argument a lot, right? I don't know why that is, but there's so many scientists who will look at the studies, and if you have a compound or you have something you've identified that will inhibit the growth of the primary tumor, they're all excited. But if you say it's going to inhibit metastasis, they say, well, that's a much harder thing. And I'm thinking to myself, well, we can take the primary tumor out. We can do chemo on the primary tumor. We can do all sorts of things on the primary tumor. What we have to stop is any more metastases.
Right.
But we also need to think about ways to target the cells at the metastatic site that have now become refractory to all the other therapies, to the hormone therapies, the chemotherapy, whatever, because that does happen. And again, understanding how those cells respond to that secondary site, how, again, this is going to require some plasticity of the cells and can then grow out, is really important if you're going to figure out how to now target the metastases in their outgrowth, right?
Right.
So,
and it's probably different. The way a cell metastasizes from a primary tumor is probably different in many respects than the way it metastasizes from a secondary, from an existing metastasis, a secondary metastasis.
And the existing metastases, dependent on where they are,
right,
may have had to respond differently to grow out because of the microenvironment they're in. Right?
It's just like, again, trying to put things in terms that are more easily understandable rather than cells and microenvironments. It's very different the way you dress, whether you live in New York City or you live where I live in Lake Tahoe in the winter, or if you live in Tahiti. And so our cancers have different environments and behave differently depending on where they are, not only depending on what subtype of breast cancer they are, but where our metastases are.
Absolutely.
And, so this is not simple. The other thing that I hear a lot is, well, they came up with treatments for HIV really easily. It's like, HIV is like a kid trying to color in the lines, and metastatic cancers are like Michelangelo painting the Sistine Chapel.
Yeah.
So much more complex.
That's an interesting analogy. I've never heard that, but very, very true. Very true.
Yeah.
So, and this is why, again, we're tackling metastatic disease from so many different angles, because I don't think there's going to be one simple answer. And I think likely, ultimately, we'll need multiple sort of therapies that you can either do together or in sequence in order to treat this. And so I think we need to understand all the different elements, right? And it's going to take a lot of us. Although I have been encouraged that I feel like more and more people are getting into research in metastatic breast cancer. I don't know if you at Metaviver feel that way, but
well, certainly based on the number of grant applications we
yeah.
get. I think back when you were getting when you got your grant, we were getting dozens.
Right, I'm sure.
Now we're getting hundreds.
Yeah.
And that, that's really exciting, because I think one of the things that is so hard is that research is not exactly a field where you're going to be driving a Lamborghini for the most part. It can be very thankless. You are constantly having to get grants to keep your lab open and to, keep, postdocs fed and all this other kind of stuff. And it can be very challenging, especially when there's that lure of, sometimes moving into, private companies and industry or a completely different field where you can make more money. So I think seed grants, the young investigator grants that Metaviver gives, that other organizations give, I think are so vitally important. And I'm really proud of Metaviver that we've been able to increase our young investigator awards to 100,000 and our translational grants to 250,000. And with the help of people watching this, by donating to Metaviver, where 100% of your donation goes into the research pot, hopefully we'll be able to award more grants and maybe even increase the amounts. I'd love us to get into the point where we're able to award grants where people are going, well, I can apply for a Metaviver grant and get as much money as I could for a breakthrough level 1 or 2 DoD grant. That would be I'm reviewing DoD grants right now, so I don't think we're gonna get up to the level 3 and 4 amounts, which are millions of dollars quite yet, but if we can do that. And I think it's so important that we continue to fund this type of basic foundational research that will eventually, not as soon as we want, but hopefully will lead to treatments that can really make a difference for those of us that are living with disease.
Absolutely. And I would say the other thing that these kind of seed grants do is, I mentioned I had a really talented student who was working on this project. She went in and she's now got a great job in industry, and this promoting the careers of people who are going to continue to do cancer research and go into all different areas. So that's awesome too, because the more people we can keep being interested in cancer and in metastatic cancer, the better, right?
Right. So again, we're so grateful for the work that you did with the Metaviver seed money and the work that you continue to do. And I hope that at some point we'll be able to see you in person, at a conference when COVID lets us. And, we're learning so much from COVID. I do want to end by asking you, what impact did the pandemic have on your lab? I know so many labs had to shut down. So many labs had to sacrifice their cell lines and their animals because people weren't even allowed to go in to do that kind of basic maintenance.
Yeah, we did have to shut down, I think, almost entirely for something like 6 weeks, but then we were allowed, and even when we shut down, if we had animal experiments going, they allowed us to keep that going. And so we had to file for special permission. And but then we had to work at a sort of reduced capacity with, spacing and social distancing, et cetera. And so there is no question that this year or year and a half was slower in terms of getting the research moving forward and generating data. And on all levels, it's been a little slower. We're finally back into it. So I certainly hope this Delta variant doesn't, because I finally feel some normalcy again. Everyone's in the lab, the investigators are back. The other thing is investigators the PIs weren't supposed to come in because we can do most of our work from home on a computer. And I have to say the day-to-day interactions you have with your postdocs and your students and really moves the science forward a lot faster, because it's that bouncing back and forth of ideas that helps the science progress. Right. And so everything has just been slow. So it's an unfortunate consequence of all of this that I think a lot of the other science has taken a bit of a hit, the non-COVID related science.
Right. Hopefully there's some things to learn, you're working with microRNAs, and the COVID vaccines were based on messenger RNA. I think there's a lot of potential there. And also, I think we've learned that if you throw enough money at something and you make people cooperate and share data,
so true.
Look at what can happen. So if we could do that with cancer research, I mean, I think a lot of us would stay not dead yet for a lot longer.
Yeah,
and I hope that happens.
I think it's starting to happen. I think there are lots of grant mechanisms that promote those kind of big projects, U54s, and I don't know if you've heard of these, but very large projects. I think it's starting to happen more and more. People are starting to put their data onto, public bioarchive, et cetera, as soon as they get it, so people can see it. I think most of us in the research community want to do exactly that. Let's all work towards a common goal, work together, share our data. There are some people who might not feel like that, but I think the vast majority do, because we're all in this to try to make a difference, right?
Right. And we as patients, we're there with you. And, I certainly hope if you don't have advocates already involved in your research that you consider doing that, because even with the basic research, and as you mentioned, when, Medivivor, the Medivivor at the time, Van, came and visited you and visited your lab, and what an impact that made on your trainees. I think there's something to be said for that. And, I firmly believe that we are stronger together.
Absolutely. I agree.
And Dr. Ford, thank you again for your time. Really appreciate it. And hopefully we'll be pulling up in our Medivivor mobile sometime in the future.
I hope so too. And thank you again for funding our research and having us, enabling us to move this forward.
Thank you.
Okay.